Hello, everyone, and a very warm welcome to Kognito Keep. Okay, get this. Did you know a single oak tree can actually sweat out up to 40,000 gallons of water in just 1 year? Yeah, it is absolutely mind-boggling. So in today's explainer, we are diving straight into the fascinating world of plant physiology to figure out exactly how scientists measure that incredible thirst. We're looking at the famous Potometer experiment. Let's jump right into it.
Here is our quick game plan for today. We'll start with a basic intro to the Potometer, check out its clever design, run through a virtual setup of the experiment, test out some wild environmental factors, and finish up with how to analyze the data and spot the limitations. Simple, right? All right, part one, introduction to the Potometer. Let's make sure we've got the foundational concepts down first.
So think of transpiration basically like plant sweat. It's the natural evaporation of water from a plant's leaves, mostly escaping through those tiny microscopic pores called stomata. And to measure this, we use our star tool for today, the Potometer, which is sometimes called a transpirometer. It basically tells us how much water a cut plant shoot is drinking up. But keep in mind, we say it estimates the transpiration rate. Why? Because a tiny hidden fraction of that absorbed water doesn't evaporate. It's actually held back by the plant to power photosynthesis and keep its cells nice and plump.
Moving right along to part two, Potometer design and the different types you might encounter in the lab. Almost every standard potometer relies on three core components working together. First, you've got the capillary tube. Think of this as a glass ruler. It has these regular little markings or gradations that let us precisely track water uptake by watching a single air bubble travel along it. Next is the reservoir. This is usually a funnel fitted with a tap, or sometimes just a syringe, which lets us push water back into the system to reset that bubble for our next trial. And finally, the end tube. This securely holds our plant shoot in place, tightly sealed off with a rubber stopper to make absolutely sure no unwanted air sneaks in. Now, scientists generally use two distinct designs. The one we're mostly focusing on is the bubble potometer, which measures the water absorbed by actively tracking that little traveling air bubble over time. On the flip side, there's the mass potometer. This one takes a different approach. It measures the actual water lost by placing a fully submerged plant on a highly sensitive digital balance. So basically, one tracks the water coming in, while the other physically weighs the water going out.
Okay, part three, conducting the experiment. This is the fun part. Let's walk through the exact setup, step by step. Before we start, we need to gather our tools. We'll need the potometer itself, a bucket of water, a fresh leafy plant shoot, some cutters, adhesive tape, and a trustee's stopwatch. But you know what the absolute VIP of this entire setup is? Vaseline. Seriously. Generously globbing on Vaseline is the ultimate secret to creating a strictly airtight seal at all your connection points, which is absolutely crucial for this to work. All right, first things first, submerge the entire glass apparatus into your water tray to flush out all the air bubbles. Now pay super close attention to this next step. You have to cut the plant shoot underwater and then quickly pop it into the rubber stopper. If you cut it in the open air, an air bubble is going to shoot straight up into the plant's delicate xylem. That breaks the continuous water column and completely ruins the experiment before you even start. Once it's connected underwater, apply your Vaseline, seal it with tape, and carefully mount it on a stand. Honestly, if you see any rogue bubbles inside the tube at this point, yep, you've got to restart the whole shebang. Assuming your setup is perfectly bubble-free, place the open end of that capillary tube into a beaker. Here's a pro tip. Add a few drops of vibrant food coloring to the water. It turns this totally invisible process into a really striking visual measurement. Next, briefly lift the tube out of the water just enough to introduce a single tiny little air bubble and dip it right back in. Note down your starting distance, hit your stopwatch, and watch that bubble travel. Whenever you want to run another trial, you just open up the reservoir tap and gently push the bubble right back to zero. Section 4, factors affecting transpiration. Because of course, plants don't live in a vacuum, right? Let's mess with the environment today. Using some really simple lab tools, we get to play Mother Nature and see how the environment shifts the results. Switch on a bench lamp and that intense light forces the plant stomata wide open, leading to way more water loss. Turn on a table fan, and the wind literally strips away the humid boundary layer of air right around the leaf, which accelerates evaporation. Now, conversely, if you wrap a polythene bag around the plant, you spike the local humidity. The air becomes too saturated, so water loss practically plummets. And finally, if you blast it with a hairdryer, the high heat gives the water molecules extra kinetic energy, meaning they evaporate much, much faster. And finally, part 5, analysis and limitations. Let's look at the math and acknowledge where things can go slightly wrong. Calculating your final result is wonderfully simple.
Your transpiration rate is literally just the distance your colorful little bubble traveled, divided by the time on your stopwatch. It's really that easy. You'll typically record this speed in something like milliliters or centimeters per minute.
But let's be real, no experiment is absolutely perfect. While the potometer is an incredible tool, it's not 100% precise. Remember that hidden fraction of water used for photosynthesis and cell turgidity we mentioned earlier? The potometer misses that entirely. It only measures uptake, not the actual loss. Furthermore, as anyone who has done this knows, maintaining a truly airtight seal is notoriously difficult. Plus, your cut chute won't remain active indefinitely. Oh, and even a slight shift in the room's temperature can cause the water inside the glass tube to physically expand or contract, shifting the bubble and subtly skewing your hard-earned data. But ultimately, the potometer proves that measuring water uptake is a fantastic window into a plant's invisible breathing process. Thank you so much for joining me today on Kognito Key for this explainer. If this walkthrough cleared things up for you, please hit that subscribe button, leave a like, and share this with anyone else who might find it helpful. But before you go, I want to leave you with this. What other hidden environmental factors do you think could secretly be altering a plant's water uptake out there in the wild? Drop your best theories in the comments below. Stay curious, and I'll catch you in the next one.